Hi, everyone. Welcome to our Spatial Omics Seminar Series 5 again. And today we are very excited to have Dr. Liangcai from University of Washington. And just briefly, Dr. Gu received his PhD from University of Michigan in medicinal chemistry. And after that, he worked at the Harvard Medical School as a research fellow working with Professor George Church, where he also worked on development of a single molecular interaction sequencing, SMI-seq technology. And after that, he started his lab in 2015 at the University of Washington as an assistant professor. And he is currently using quantitative protein interaction profiling and other sequencing technologies to shed light in biology and biochemistry. And without further ado, I'd like to actually turn over to them, Dr. Gu.
Hello, everyone, and thanks, Ahmet and Rong, for inviting me. It's very exciting to interact with this fast-growing and dynamic community here. And today, I will introduce our spatial barcoding method called PIXEL-seg and analysis of the chronic pain-regulated transcription in the brain. And as a self-introduction, I want to tell you what my lab is doing. My lab focused on protein engineering and the spatial barcoding and sequencing technology development. And we are very interested in making two types of tools for in vivo studies. First, genetic encoded biosensor, such as GCAM, for real-time monitoring and actuation behavior animal. We are also making sensors for drugs and other biomolecules to achieve the high spatial temporal solution. We're also developing the barcoding method for three-dimensional multi-omic profiling with a single cell, even and subcellular solution. Our goal is to integrate these tools to study the in vivo cell functions and their omics changes. And today, I will focus on the second tool. So in terms of the biomedical application, my lab is particularly interested in the function or activity regulated gene and changes in the tissue. And some cells involved in certain function can get lost and during the traditional tissue dissociation analysis. So we're trying to use our spatial assays to avoid that. And second, we are interested in the cell-to-cell communication, which definitely requires spatial information. And we're also interested in applications. In precision pharmacology, we want to understand the drug action in vivo. And I'm interested in the pain mechanism, like neuropeptide modulation, and how to develop painkiller drugs to avoid addiction. We're also interested in mapping senescent cells in mouse and human tissues and how their secondary phenotype interact with normal cells. And the goal is to guide the development of drugs or immunotherapy to remove these cells from our body. And today I will give you an example of the PIXEL-seg analysis of the pain-regulated gene expression in mouse brain. So my talk has two parts, the biology part and the technology part. And first, I will give you some background on our PIXEL-seg development, and then our recent efforts to push the technology to the single cell analysis. In the second part, I will tell you our analysis of a very important brain region called parabrachial nucleus, and how the cells in this region respond to chronic pain treatment. Before talking about PIXEL-seg, I want to explain my understanding of the trends in the single cell and spatial RNA sequencing technologies. And there are so many new technologies developed in past decades. And I just chose a very few examples for comparison. I searched the Google Scholar citation counts for some original papers to understand the needs in this field. And so this one is the proof of principle paper and for CGRP, and people use the culture cell and to do the demonstration. And this one is a drop-seq. It represents the technology maturation allowing the wide implementation of CGRP and using the tissue dissociation. At a similar time, spatial RNA-seq technologies such as PIXEL-seg, morphologies, spatial transcriptomics showed up and grew very quickly. And you can see their citation increase and is similar to the proof of principle paper, but still much slower than the drop-seq. And we understand the spatial information is very important. So what we are still missing here? I think we are still waiting for this non-dissociative single CGRP with a true single cell resolution. And this technique needs a high precision and sensitivity comparable to the standard dissociative single cell RNA-seq. And it should be widely used by end users. So we can test different types of biological samples very quickly. And it should be scaled for both the small size or large size tissues and also now high throughput. So this is my understanding. And generally speaking, spatial and transcriptome profiling technologies are based on two major strategies, and multiplex imaging and spatial barcoding. For imaging-based method, the R detection can be achieved by in situ sequencing. Here I just used the PIXEL-seg and developed by the Church Lab as an example. It can also be achieved by multiplexed hybridization. I used the morphologies from the Rong Lab as an example. The other strategy is based on spatial barcoding using oligoarrays to induce the spatial indices into the synthesized cDNA on the array. A good example is the spatial transcriptomics technology, and now sold under brand name Visium by 10x Genomics. So by comparison, the imaging-based methods are superior in spatial resolution. They can also achieve high RNA capture efficiency by doing multiplex probings or isotropic tissue expansion. The barcoding-based methods have much lower spatial resolution, but they allow the global naive transcriptome profiling, which can greatly facilitate some biological discovery. Importantly, these barcoding assays are more friendly for end users. And this year, a few groups, including mine, made breakthroughs in the resolution and capture efficiency. Resolution can reach to 0.5 to 1 micron. RNA capture efficiency can reach 10 to the 4th UMI for regular size mammalian cells. And I have been working on the polony people called cluster amplification and sequencing since 2010, starting from my postdoc trainings in the Church Lab. My lab has been working on testing like different polony amplification conditions, such as different gel structures, the primer and template designs, and different cluster densities. So we're trying to make this poly gel manufacture process better and faster and cheaper. The idea of using polonies for spatial barcoding has been there for a while. But there are some challenges to create the high quality oligoarrays from polony gels. So we testing the different amplification conditions. We understand these clusters can grow into different weird things, and they can cause issues for this final oligo formation. So we proposed the PIXEL-seq method several years ago, and it took us a while to make it work efficiently. We find the right and the gel condition and also the cluster and density and intensity. And here's how it works. The first step is to make the cross-linked polycromate gel, and we can randomly seed the template on the gel surface. And then they can grow into the polonies or clusters, and they can train this double strand and DNA by restriction digestion to get the 3' adapter and sequence exposed. And then we use our in-house built sequencer to sequence these polonies. And we thought about using the standard Illumina and flow cell for the sequencing, but our gels have certain features making them not compatible for the standard Illumina sequencing. So we have to use our own sequencer, and then we can add a different capture and sequences and to the 3' of oligo to do to capture different targets. Once we finish the manufacturing, and we can put the tissue on the gel surface and then do in situ cDNA synthesis. After getting this barcoded cDNA, we can amplify them and make a library for next generation sequencing. Once the gel is manufactured, the assay can be done in a few hours. Our polony gel is similar but different from Illumina flow cell. And we found the polony grows on the surface of certain cross-linked polycromate gel can form a continuous structure instead of standard peak-shaped cluster. So these two flow cells were amplified under the same condition, but using different gel structure. And so for this kind of gel structure, you can see this kind of like a peak-shaped polonies. The DNA are not evenly distributed. But for our certain cross-linked gel structure, we can get a continuous polonies. Although these polonies are connected to each other, we still can find a boundary by DNA sequencing. By taking advantage of this phenomena called polony exclusion effect, we can generate an evenly distributed oligo array, which is impossible for the standard like oligo printing or other method. So this kind of cluster is very suitable for the tissue mapping. You always get a continuous image of your tissue sample. We further developed the pixel level and base calling method to identify the polony centers and their shapes. Continuous polonies often have irregular shapes, so we need to extract intensities from all pixels to do base calling, and then use coordination based in the barcode correction to correct the base calling errors. Basically, we try to identify the major barcode species in each pixel. Currently, we're still trying to improve our imaging setup to allow the sequencing of the higher density polonies. By doing so, our resolution can reach 2.5 micron. As I just said, the polonies are on the surface, on the gel surface, and this gel can largely restrain the template diffusion even without the tissue fixation. To test this, we used the mouse brain sample without fixation, and then we used the Sci5 and DNTP and to enable the cDNA. We can easily achieve this and single cell resolution. If the nucleus showed up, you can see a dark hole in the middle of these cells. Like other spatial barcoding technologies, and we used the mouse olfactory bulb and to test our technology performance. So this is the total and UMI and density map showing this continuous structure details. And to quickly access the data quality, we just simply use the regular and sized bins to randomly group transcript reads for unsupervised clustering. So we can easily find the 10 major cell types grouped into different layers. Of course, the many genes showed the spatial and gene patterns very similar to the in situ hybridization data. This spatial barcoding method shows the higher detection and specificity. I can see in the in situ second data, in situ hybridization data, you can see a lot of these these cells in the inner layers can get detected. The gene get detected, but in our case, they're not there. So this tells us that this sequencing-based method can have a better specificity. And we compared our data with other mouse olfactory bulb data generated by other spatial barcoding and technologies. And we can see we can get a very good resolution and also have a very good capture efficiency. And so given our improved data, we asked ourselves, can we do the true single cell segmentation instead of random bin picking? Actually, the mouse olfactory bulb is not an easy sample to do that. But we don't want to pick sample. We just we start from the olfactory bulb. We want to continue to do this. We just want to do this segmentation on this top tissue, because this can provide us a rigor test. My lab is working on 3 single cell segmentation method. The first is commonly used one, the standard cell image guided segmentation. Available segmentation method developed by other people are often based on deep learning. So you have to do a few rounds of training in order to get a reasonable result. We test the several methods from other groups. And my student, Nick Selpals, published early this year. And he can identify a large percentage of the cells from the human heart and liver tissue images. The second method is to combine the Protein-PIXE-seq to the RNA-seq to find the cell boundaries. And here I used a human heart tissue as an example. As you can see, the DNA tagged WGA allow us to identify the cell boundary similar to the HE staining. And for this sample, we just sequenced this small number of reads by MySeq. And so you can see these boundaries are not very continuous. But we found the sequencing depth was very low. And if you get more reads, we can definitely see a very smooth continuous boundary, and this will allow us to do a very good the cell segmentation. However, these two methods are not okay for the mouse brain sample. They are very straightforward, but they're very difficult for brain tissue sections such as the olfactory bulb. And as you know, the brain cells are highly diverse with distinct morphology. So finding the cell boundary is much more difficult than other tissues. So this is nasal staining showing all cell nuclei. You can see some regions have a very high density cells. And even for some low density region, if you look at the EM image, and you can see there are more cells there, and with kind of weird shapes. And some neurons were sectioned of their dendrite or axon region, so you cannot see their nuclei. Although it's very difficult to use one or few staining methods to show all cell boundaries. So staining methods are not some staining methods are not compatible with our assay. Additionally, sometimes a stained cell image could be misleading. You see the cells there. You see cells there, but you might not detect the RNA released from this cell because this cell probably stayed in other nerve and far away from the section side. Even you make a very thin section. So this cell, the tissue thickness will cause some misleading for this imaging guided segmentation. And so my postdoc Xiaodan proposed a very small ways to do that. The third method based on this direct reads and based segmentation called v-seq. So basically, we generate the weighted network from this UMI density map by considering the UMI density in each barcode, their distance, and the shared genes between the adjacent barcode. And then we can cart, and then we can rank these edges, and we cart the no-score edges, and to segregate the transcript reads into the single cells. And this method works efficiently for this mouse brain sample. It starts from this U density map. This is a segmentation result. You can see, and there is the very good coordination. And if you look at some marker genes, you also can find some coordination. After the segmentation, we got 27,000 cells from one olfactory bulb section that can pass the filter. Importantly, and we can get a large percentage of the mapped UMI, and that can be segregated into the single cell with sufficient UMIs for the single cell analysis. So this part I like the best because we try other approaches. Sometimes we're gonna lose a lot of mapped reads. So Xiaodan's method can allow us to get all the to use most of this mapped reads for the single cell analysis. So the 23,000 cells were annotated into 25 cell types, which can nearly grouped into 6 layers. And we found their cell spatial locations are the same as the reported in other papers. So so far, the mouse olfactory bulbs has been analyzed by different methods, but these are data. This provide the first high quality and single cell spatial analysis with this true single cell resolution. We're very proud of this. And to demonstrate that PIXEL-seg is true, non-dissociative and single cell RNA-seq, fully dependent of this dissociative and single cell RNA-seq. We compared the cell annotation results from the single cell RNA-seq-guided and annotation and totally unsupervised clustering. So this is the single cell RNA-seq data guided, and this is the unsupervised clustering. You can see the results are very similar, patterns are very similar. By comparison, if we use the random segmentation with the 10 micron beams, and we also did by doing this single cell annotation guided single cell data guided annotation and unsupervised clustering. So this data still okay. You can find the layers, but the data quality is not good as the RV-seq and results. For this unsupervised clustering, and most of the layer information get lost. So this result tells us that PIXEL-seg coupled with v-seq can be independent of this dissociative RNA-seq to get a high quality single cell analysis. We next look at the relationship between the segmented and cell sizes and the UMIs. As we expected, the bigger cell always give you higher UMI counts. However, some cell types are bigger than others. For example, the PGC cells, and the average, so the peak of the size distribution is close to 20 micron, but the MTC cells is lower than 10 micron. And this data analysis showed us if you have the 20 micron cells, we can get a UMI close to 10,000. So it's very similar to the dissociative and single cell RNA-seq. In addition to cell sizes, we also look at the UMI densities in different cell types. Yeah, some cells like PGC have the highest density than the others, and MES and OEC cells have the lowest. So this is quite interesting to us, and can help us to do these reads and based cell segmentation, because we want to use this reads density to separate different cells next to each other. And next, I will talk about our analysis of a very interesting brain region called brachial nucleus, or PBN, and the chronic pain-induced changes in this region. We chose this region for two reasons. First, it's critical to many basic brain functions. Second, its anatomic structures with this kind of scattered cells is very common in the brain, but many spatial technologies try to avoid these kind of structures. They prefer the near brain structures. So for this kind of like brain region is very important. They are very important, but it's very difficult to dissociate and isolate some no abundant cells from this structure. They are biologically important, but they can easily get lost during dissociation. So we want to demonstrate PIXEL-seg can be used to analyze this type of tissues, and then there will be numerous of applications. So where is the PBN? In the mouse brain, it's in pons, next to the cerebellum. So why it is important? In simple words, it follows the homeostasis of the whole body. Some people think it represents the basic forms of the consciousness. So basically receive sensory inputs from the peripheral, such as taste, ingested behaviors, pain, atomic controls, and it also talks to foreign brain structure to mount responses to counteract threats. We can expect that this neuron in these PBN regions are intensively involved in electrical and biochemical computation. So this is a very great samples for different spatial technology to try their potentials. As I said, this region represents a very good example of the common brain structure and different from near structure. It consists of relatively compact clusters of the neuron cell bodies called nucleus. Our brain have hundreds of these kind of structures. By looking at the lethal staining, we already know it must have very diverse cell population. And it also have some sub-regions here. Probably going to have the different type of cells. Here is the typical UMI density map of a PBN section. The section size is around 2 by 2 square millimeter. This map contains the 31 million UMIs and more than 27,000 genes. And we can clearly see anatomic features based on this map. You can see there are some big cells and small cells. These bright dots are corresponding to the cell bodies. By eyes, we can draw this PBN boundaries. It has 2 fiber tracks, and this one, and this one. We can clearly see the pattern gene expression by only looking at individual genes. So now it seems that our data quality looks fine for this type of tissues. Because the cell types in these regions are not well understood. So we first try to perform a 3D analysis by sampling 3 coronal sections at different levels, anterior, middle, and posterior, and each have 150 micron distance. So this is a total UMI and density maps. We also look at some favorite marker genes of some neurons suggested by our collaborator Richard Pumpture. They have been studying the CGRP neurons in the PBN region for more than 80 years. These neurons express the CACA gene, CACA neuropeptide genes, and they're more located in this middle section in this region. They are kind of tightly crowded in this small region. When you do in situ hybridization using some marker gene probes, they look kind of like a homogeneous. It's very hard to separate individual cells. So we want to try our technology to analyze these neurons. And next, the olfactory bulb, we did a single cell segmentation by V-seq. The unsupervised clustering identified 18 clusters showing specific spatial patterns. So we think these 18 clusters should represent some cell types. But I'm not going to go through all these cell types. It's going to take a very long time. We're only going to focus on the CGRP neurons in the PBN region to answer our collaborators' question. So here are these CGRP neurons. The first question and our collaborator asked is, do CGRP neurons have different cell types? Based on the shape of this cluster, the answer seems yes, because they are kind of like separated into 3 clusters. So we search for the marker genes, and interestingly, they differentially express the 3 neuropeptide genes, the TAC1, CACA, and NTS. So this confirm, and they at least have these neurons at least have a 3 and cell types. And the second question is what kind of do these neurons have any spatial organization in the PBN? Here, we highlighted the segmented cells in these 3 small neuron clusters, and the color and their cell types. And so you can see here, this green neurons and the CACA ones are kind of surrounded by the blue one, the TAC1. There are a small number of the NTS neurons located in the middle of this structure. So if you look at these 3 sections, and there's yes, it looks like there are some spatial patterns. So we believe these neurons must form some spatial organization. So this data provide us the first evidence of this spatial organization and cell types of these CGRP neurons in the PBN. Of course, if you look at the distributions of these the segmented CGRP neurons, and in the middle section there are more TAC1 neurons there, and yeah, my collaborator is highly interested in this kind of neurons because they have so basically they can be respond to like many behavior such as conditional taste version, pain, and many other threats. The third question is what kind of transcription changes can happen when these neurons are activated by certain behavior, such as pain? Our understanding that when the neuron fires, the calcium concentration change can activate some kinases, which can cause the activation of some pre-existing transcription factors, such as TRAP. They will immediately translocate it into the nuclei to activate a small set of the immediate early genes, such as FOS. These early genes will be further activate cell type specific innate response genes, which have been fully understood. There are like 300 to 500 innate response genes. So far, there is no good studies to fully analyze this gene and transcriptions. This gene expression will further trigger cell communication for cell adaptation and modulation. So we want to use the PIXEL-seg to understand these changes. So this is kind of like a very open field and to allow us technology to test its power. To answer the sad question, we used the chronic pain mouse model to analyze the gene regulation in the PBN. The mice received the sciatic nerve ligation surgery and waited for 5 or 30 days before taking the brain sample, and we do flash freezing and then cross-sectioning before the PIXEL-seg assay. The assay itself takes half day. And after we make library, and we use Nova-seq and for the data analysis. So interesting, we found many changes by comparing the naive and treatment mice, and Sheldon only listed some top genes here. So these genes are going to take some time for validation. So we're doing a validation using other techniques. This is going to take a while. But here I just want to show like 4 downregulated genes. The first 3 are neuropeptides: the KAKA, PANK, and SG2. And the 4th one is STEM1, which is the inhibitor of microtubule formation and reported to control theory. Very interestingly, these genes, including the opioid peptide PANK gene, were downregulated. So these neurons project to several forebrain structures. So to reduce the production of the neuropeptides, we're going to change the modulation patterns in these regions. So here you see this SG2. So SG2 is kind of like induced by this this pain behavior. This result remind us the very interesting discovery from the Greenberg lab last year. They found the neuroferrin induced the early immediate gene of phosphoprotein. And they can activate the transcription of the SCG2 gene encoding multiple neuropeptides. The story is like that. The CGA and pyramidal neurons is important for long-term memory, but they are inhibited by PC and CCK neurons in naive mice. Exposing the mice to neural environments can activate the specific CA neurons, and then result the FOS expression. The FOS expression activates SCG2 encoded peptide expression, and then that's modifying inhibition strengths of the PV and CCK neuron. So long-term memory can be consolidated. So in our case, the chronic pain treatment decreased the peptide expression. So our guess is that less peptide sent to the forebrain regions will mount decreased threat responses in the chronic pain animal. Currently, we are digging into our data to answer the 4th and the 5th question. We wonder if the different CGRP neuron types or individual neurons show different transcription regulation. So we're trying to look at the individual neurons and their neurotypes in each spatial location to detect their transcription changes. We also ask if the pain treatment can induce or interrupt the local communication with other cell types, such as microglia, astrocyte, oligodendrocyte. So this analysis lead us to analyze the cell locations and their subcellular morphologies. So I have to say there are many things to be developed for the data analysis, and we are open to collaboration on this. At the end of my talk, and I want to mention the subcellular solution for our data analysis. People in the field think currently some subcellular data analysis is not fully utilized. I totally agree. For our data analysis, we can see subcellular morphologies of some cells like astrocyte, microglia, oligodendrocytes. And we believe these morphologies will be very useful for cell-cell communication analysis, as I mentioned in the question 5. Finally, I need to thank my people. And we have a small team, but the people are very talented and hardworking. And we built the whole thing from scratch and been through all sorts of troubles in the gel fabrication and DNA sequencing, the PIXEL-seg assays, and the data analysis. I feel I can learn new things from my people every day. My staff engineer, Lisan, has been working on the polony and jump application for 6 years. And Xiaolan, my postdoc Xiaolan, has been developing this assay and data analysis. Our graduate student, Runze, has been working on the single cell segmentation and the statistics. So we're working with the Palmature lab on the mouse brain samples, and 2 physicians, Shen and Ying, on the human tissues, and also be involved in our data analysis. And thank you for listening.
Yeah, very cool, Liangcai. Congratulations, it's such a amazing technology.
Thank you, Rong.
Yeah, so I think if anyone has question, you can just unmute yourself and ask. If you want to use the chat box, that's fine too. But we try to keep this kind of more informal, more interactive format. Feel free to unmute and ask.
That was a great talk. I wonder if you could give us a sense. Oh, sorry, this is Silas Miniotis. I was wondering if you guys could give us a sense of how, what scale of sequencing is required for PIXEL-seq. For a given area, how many reads do you need to achieve, to detect the the number of UMIs that you presented here in this talk per cell?
Yeah, so for our mouse samples and the PBN of factory bulk, and for like 2 by 2 square millimeter sample, we normally need like few hundred million reads. And for a factory bulk, I think we got so our mapped UMI is over 100 million. So we got I think 400 raw reads. And for the PBN, each section we got around like 32 and 40 million mapped reads, and we need like 200 to 300 million raw reads.
Okay, great, thank you. And just to eliminate confusion here, a couple of people on my team, I think, are logged in with my Zoom information here. And so this is my colleague Kunal asking a question in the chat that I'll vocalize for him, which is, he's asking if spatial barcodes are located on the surface of the polony gel or throughout.
Sorry, on surface. Yeah, it's on the surface of the gel.
Thank you very much.
Yeah, yeah. So this is very important, and we trying to avoid putting the DNA cluster, these polonies, inside of gel. So that can cause the no capture efficiency. Even you can see very good sequencing signal, but they are not efficient to do these RNA capturing.
But yeah, how did you how did you do that? So since the gel is kind of 3D porous material.
Yeah, we have to control the gel structure, pore size, and we also have to control the template size. So your template won't drop into the gel, and they're gonna have more open space to the applications. Most of this double-strand DNA will be presented on the gel surface, and they can get exposed to the tissue RNAs.
Cool. I think Edward raised the hand. Do you want to just unmute and ask?
Hey. Yeah, so I'm actually at the University of Washington as well. I'm in the biostatistics department, and the lab I'm in, we've been working on developing methods for spatial transcriptomics, and mostly just focusing on the 10x genomics technologies. So I don't really have an exact question, but I was wondering, is any of your data for PIXEL-seq publicly available? I think we'd be really interested in like working with some of your data and seeing we could like develop methods for it as well.
Sure, we can talk about it. And at this moment, because the paper is not published and fully officially published, so we haven't posted data to some servers. But for some collaboration, we can consider sharing the data.
Okay, cool. Yeah, we'll reach out then.
Yeah.
I had a question I was hoping you could answer for me. I was curious
Alex.
about, yeah. So, of course, to expand this ultimately, I think a lot of people would be interested in how you would assemble the spatial barcodes without the complicated machinery that you might have. So is there a sort of, I don't know, like a microfluidic strategy or a light-based patterning strategy that you could use to assemble the spatial barcode, like on a benchtop?
Very, very good question. We the SLICE-seq and other and similar methods, we always need to do the sequencing, and we're thinking about they might have some strategies to avoid doing the DNA sequencing, and we are starting working on that. So if you want, the answer is yes, potentially yes. We need to find, and so we try very hard to generate this kind of gel for this tissue mapping, because we think this gel have the potential to avoid the sequencing, and sometimes you'll find a way how to transfer the DNA from one gel to other gel, and then this will provide some solution for a no cost gel. And we think the current kind of Illumina and clusters, and they're on some kind of linear polychromate gel. They're very hard to do this because the mechanic force is not very good. So we're trying very hard to find a very good gel structure to make the whole manufacturing process faster, cheaper. And we hope this gel the manufacturer will be close to a kind of printed array and even have lower cost.
Yeah, what I can imagine is you can use kind of one gel as the template and kind of stack another gel on top and just transfer.
Yeah, that's one solution. But this need some certain technology development.
Yeah.
We believe our gel have the potential to do that.
Yeah, this is very flexible, and also you can do all the manufacturing and just send a gel to the kind of collaborators, right? So
exactly.
After you do that, your collaborators don't have to. Yeah,
exactly. We hope we'll get we'll show some results very soon. Then this idea have been there in our lab for a while. We already start to work on that.
So I think the 10x spatium requires some kind of tissue digestion optimization, permeabilization optimization, something like that. And do you need to do that? Or
so right now we do this this tissue mapping and in a similar way as the slide-seq, but we definitely can do fixed tissue, and we because for academic research sometimes we don't need we don't have to do fixed tissues, and there's some fresh sample we can easily get, and we don't need to get this kind of like human tissues and in kind of fixed condition. So we focused on the fresh frozen tissues. This allows us to get very good data and also good in resolution.
Yeah. I think my question is, I remember I think a THX technical specialist told us you need to do the tissue permeabilization optimization first before you run your real sample, right? So they give you like a dummy array. You look at like 5 minutes, 10 minutes, 15 minutes, maybe so that's to remember that until you say, okay, you get a very crisp signal on a dummy slide, and then you use that condition to do your Bayesian experiment. So I wonder whether or not in your gel substrate you have to do that as well.
And sometimes we have to do the test. We just use the unsequenced the gels to do the test and make sure we use nanomyseq and to do some quality control, make sure and the reads qualities are good, and then we start to use the sequenced gel. But normally we don't. For our experiment, once we know the gel manufacturing and condition is fine, we don't need to do the QC for different tissue samples. We did, but not a lot.
Okay.
One more question. I was wondering if you could play with the gel like cross-linking density to do any expansions and sort of resolution increase that way.
That's possible. And let me think. So in that case, you have to bury your tissue into the gel, and then you have to you have to do the amplification and in the middle of the gel. Yeah, I think I would say that's possible, but I don't think at this moment we have the leads to do that, and because we already get pretty good UMI numbers in very small cells. So,
I just imagine in 5 years you'll call it super resolution spatial transcriptomics.
Yeah, we can think about that.
Thanks.
Yeah, I think also your cell segmentation pipeline was super impressive, and that's not easy. That's super challenging, even for just a fluorescent imaging of the cells, and you do segmentation based on just the face, right?
Yeah.
So that was done with the brain tissue side.
Yes.
Okay, so in that case, I remember I think the fish folks that showed the image and the segmentation of the neuronal cells, they were pretty much able to just segment the cell body or just the nuclei. But it seems like you can see the more complete cell shape. Maybe the cell type is slightly different, or your algorithm.
That's correct. So right now our segmentation allows to find some weird cell types, and I think most of them could be true, and probably we still So I believe we still have certain percentage of the cells probably get mixed barcodes from different cells. But at least it's working for our single cell for our cell clustering, and the result looks fine.
Yeah, I'm actually very intrigued by this result, because you may have the chance to really see where you have some kind of region specific transcripts. Okay, for example, localized in a dendrite, localized in the axons only, but not in a So about the
Actually, we are working on that.
Okay.
Yeah.
But have you looked at like the kind of nascent RNA versus the more mature RNA and the spatial distribution within the cell?
And so Xiaolan started to do that, and they look at different isoforms, and there are too many analyses to be done, and we just need so right now we have to you have to make some direction, see what kind of phenomena or what kind of like a biological discovery we want to make. So yeah, the PBN sample we just get started and to do this kind of the isoform and subcellular and morphology analysis. You're going to take some time.
Yeah, I think one question from Collis Price. Do you want to ask yourself? Or I can.
Sure.
Yeah.
Great talk, by the way. I was.
Thank you.
So this is my lack of knowledge more about, I guess, SlideSeq. But I think I was talking to another colleague, and it seems like the principles of the technology seem a little bit similar. So can you talk a little bit about some of the advantages that your analysis in PIXEL-seq might have over SlideSeq or other types of technologies?
Yeah, so we believe our technology will allow us to move to the true like non-dissociative and single cell seq.
So do you think in terms of, I guess in terms of limitations of PIXEL-seq, I thought the cell segmentation was really interesting. Was some of the like how you guys were approaching cell segmentation. Do you see that as the bottleneck, or do you think it's more about, as you said, kind of getting that? I think that you called it uniformity of the. Like I would be curious to hear what you think the potential bottlenecks down the road might be.
And so of course the our cluster is still it still kind of have certain size, and I would say we're still gonna reach to our spatial resolution. So we always going to get some cells with some segmented cells with mixed transcripts from mixed cells. So that's our limitation. I don't think this can be easily solved in near future. But we can push to the limit. But I think the current advantage for this type of method it allows us to detect some like rare cells and rare functional cells in the tissue, which might get lost in a dissociative and scale-seq.
Yeah, definitely. Thank you. Great talk.
Thank you, Celeste. You raised your hand.
Yeah, sorry to pepper you with questions. I mean, obviously, I'm very interested in the technology. So following up on this cell segmentation thing, I agree this is very interesting and is something that actually differentiates this from SlideSeq, because SlideSeq, to my understanding, it's incompatible with imaging generally. But you alluded to certain stains being PIXEL-seq compatible. Is that just because you had DNA barcodes on WGA and not other things, or are there certain dyes that actually won't work with the acrylamide gel or something of this sort?
So I would say all the dyes working for the SlideSeq working for and the VZM, and we're going to work for PIXEL-seq. I mentioned that some dyes may not going to work for PIXEL-seq because we realize are certain some cells are probably going to use very unusual dyes to see the boundary, and that kind of staining will not going to work, and might not work for the PIXEL-seq. We haven't tested many dyes, but we know some dyes are going to decrease our capture efficiency.
Thanks.
Yeah,
So,
I think 2 more hands raised. So, yeah, you can go ahead and unmute yourself and ask them Perot and Sutini.
Oh, thank you. Hi, Dr. Gu. That's a great talk. I was just kind of curious about the human tissue that you have on the screen. I'm curious that, because the PBN is at a very difficult anatomical locations at the pons and also at the midbrain. I'm not a neurosurgeon. I was just curious what kind of human tissue you get and why the patient want to donate the tissue, if you don't mind to comment. Thanks.
Yeah, we have the heart and lung and liver tissues from physicians, and so these doctors and they're doing transplantation, and they can provide some diseased and the human tissues and some could be some healthy tissue and healthy and human tissue, but that's not easily available. So we but we are thinking about soon we're gonna test the fixed human tissue, and so the human tissues and right now we mainly we mainly get them from some collaborators.
Is it from autopsy?
No, from transplantation.
Wow. Okay.
Yeah, and Peril, probably ask the question.
Dr. Gu, thank you again so much for the talk. Great information. This is probably an appropriate question for the last question. I just wanted to know, is there any publicly available information about the PIXEL-seq process that is available to the public?
We're going to try to release all the details, and once our paper gets accepted. But if you want to set a collaboration, and we can talk individually.
Okay.
Yeah. This manufacturing is not trivial, and we were thinking about working with industry and partners to make this gel widely accessible, and it's gonna take some time.
All right, understood. Thank you.
Thank you.
Great. And in the meantime, one question from here. I think we are almost done here. If anyone has questions, of course, please feel free to unmute yourself. So the question will be about the biology, right? The transcription. You're suggesting that there are waves across these brain regions, right? Cell type specific. So how about the proteomic signature? Because transcription is prior to that signal, right?
Yes.
In the dogma. So what are your thoughts on this? Like maybe your methods can be adaptable to protein screening in parallel?
Of course. And we're already prepared for that for a while. And yeah, so my lab is in the Protein Design Institute. Most of people and our labors are working on proteins, and my lab also developed all sorts of this kind of binder, a mini binder and screening method. We can generate the binder for different antigen very quickly, and we think this binder will be more useful than the traditional antibodies. If antibodies, the size still kind of big, and if you use mini binder, you can detect the different epitope of the binder, so you can detect the protein interaction, protein modifications, and you can get a higher UMI because the antibody is too big. They'll make this environment very crowded, and if you use a small binder, you can see more detailed protein details and using by doing this kind of multiplexed the binding. So we already have these kind of mini binder libraries, and we start to get this DNA tagged binder for some initial test.
Yeah, that will be exciting, especially if they even work at the same time.
Yeah. A lot of things. I would say that the protein omics will be more exciting because you're going to see more subcellular information. So they're going to strongly affect our spatial analysis.
Exactly. And another simple question is about the segmentation. Have you guys looked at the false positives, and how would you eliminate those?
And sorry, so since
false positives, like if you are capturing a cell and a dot, and because there's looking at nearby distributions, right?
Yes.
If you're picking something from nearby cell or not, like, do you have a way to? I mean, it's a great method, but I'm curious about limitations.
Yes, we definitely have the we're definitely going to have some cells and from this like mixed some segmentation from the mixed cells, and we right now it's very hard to assess the ratio. We just look at the clustering and the unsupervised clustering results. We just want to make sure, and if we're going to have like a big cluster and that cannot be well separated. Then so I would see we do have that that population, and but we still can further improve it. We can do some kind of like transcript analysis to find a better boundary between these adjacent cells, and yeah. So this still we still need time to develop and this analysis.
Actually, your sensitivity would be good if you stain, for instance, something like poly A, right?
Mm-hmm.
And you could color the entire cell maybe, right? And
yes.
then that could be a maybe exact mask.
Yeah, yeah, that's possible.
Right. Yeah, I mean, like something really covering entire thing. Great. Any other questions from the audience? If not, let's thank to the speaker again. Thank you, Dr. Gu, for this exciting and fantastic talk. And
bye-bye.
Clapping. And before we wrap up, I'd like to announce that also we have the last but not the least speaker of this seminar series, Dr. Douglas Shepard from Arizona State University. And he will be talking about more imaging approaches, versatile, high resolution, and large field of view oblique plane microscopy for spatial omics. And after that, we'll be starting to plan for seminar series 6. And if you want to nominate any person, please let us know. And if not, let's wrap up for today. Thank you guys for participating, and hope to see you guys next week, and have a nice weekend.
Thank you.
Thank you.